Welcome everybody to the HRI seminar series for October. I wanted just to start by acknowledging that UMBC is located on the traditional territory of the Clay Dene. And if we all just want to take a moment and reflect a little bit on what that means for our lives to live here in these lands and how fortunate we are to do so. So this is to introduce the HRI Seminar Series Planning Committee, myself, Dr. Ferber, Leanna, and Bree, who do a tremendous job organizing this all, as well as Dr. Anna Robinson and our student member, Nizia Shakar. And of course, our financial contributors, which is Sun Life and the Faculty of Human and Health Sciences here at UMBC. So just a reminder that this was up when you guys were eating and entering the room here. This is a new portal that has information about different HRI members, where you can go to connect, learn about what kind of research is being done and who you might want to collaborate with. And for upcoming events, so we have a visiting. Oh, we have lots of things going on. Busy fall. It is. But our two seminars coming up, we have a visiting scholar, Jackie Hughes, a nephrologist, and as well as Dr. Brian Christie, neurobiologist, coming in November. So for today's presentation, I am excited to welcome Genome BC here. So Genome BC is a nonprofit organization supporting world-class genomics research and innovation to grow globally competitive life science sectors and deliver sustainable benefits for British Columbia, Canada, and beyond. So I am going to let our guests today tell you more about Genome BC and their initiatives. So I would really like to welcome Zsuzsanna Hollander, who is the Director of Data Science, and Annaïck Charles, the Research and Innovation Manager for Data Science. And I am told that they are going to tell you a little bit more about themselves. So come on up.
I'll let you come up. Yeah, we can just, oh, sorry. You can just share. I'll just get started here. Oh, is that good? And you can probably turn off your camera if you don't want. Sorry, I'm just, now that my slides came up, I can no longer see the Zoom. Just a moment. I got it. Stop sharing. Oh, it's off. Okay, thank you. That's why I was like, I didn't click it yet, but it disappeared anyways. It's like, it's a mind reader. All right. Thank you so much for all of you for coming. It's nice to see that the room is actually filled up. It's lovely. It's a pleasure to meet you all. And so Annaïck and I are part of the data science team, which is fairly new at Genome BC. When our current Chief Scientific Officer, Federica Di Palma, joined Genome BC about 4 years ago, she wanted to put data science at the forefront, knowing that the genomics data that we're funding is data, right? And so data science is behind all of the sectors that we're working on. And so she wrote up a data science strategy and then hired me to implement it. And then about a year later, I was hired to help with the metadata commons that we'll be talking about in just a little bit. Both of us have our experiences pre-Genome BC in health research. We both worked in 10+ years in that area, working with data analytics. And so we would also like to acknowledge that we live and work on the traditional unceded territories of Canada's Indigenous people. Specifically, our offices are located on the territories of the Coast Salish people, including the Musqueam, Tsleil-Waututh, and the Squamish nations. And so just a little bit of our intro of who we are. We sit at the intersection of government, academia, and end users, such as industry, that we work with. And our mission is carried out through different tools, such as thought leadership, convening, partnerships, and one of the things that you may be interested in, funding programs. Our aim is to move genomics from scientific pursuit to be useful in everyday life. And hopefully, we have now been in existence for 24 years. So next year, we're celebrating our big 25th anniversary. And through that time, there has been quite a bit of progress in terms of technology when it comes to omics. So for us, genomics means not just specifically genomics, but all the different omics, transcriptomics, proteomics, metabolomics, as well as the data analytics that come with it. And so there has been a lot of progress in each of those areas, as well as how we view genomics for the different sectors that we're supporting. And so Genome Canada is sort of the federal level of the genomics enterprise, as we call it. And a lot of their funding is coming from the federal government. And then we have 6 regional centers that are independent that cover Canada from coast to coast. We support not just the health sector, but a lot of the other sectors in the agri-food and natural resources area. And now that we have a data science, we also have a nice purple square or a circle with to indicate that we're also supporting the community and supporting collaborations in the data and technology area as well. And so I figured before we jump into discussing the metadata commons, which is unfortunately or fortunately, depending on how you're looking at it, I love talking about metadata. And so I didn't want to get into it. I wanted to first start about funding opportunities that you may be interested in now or in the future. You could keep it in mind. One of our funding programs that we have periodically, so 3 times a year, is called GeneSolve. This has been redesigned last year from what it used to be slightly. So the redesign, the goal of the program are similar still, as in if there is a sector partner, which could be for-profit, not-for-profit organization, it could be a health authority, it could be the ministry, it could be any number of organizations. If they have a problem that could have a genomics or omics-derived solution or omics-enabled solution, they could work with academic partners to solve set problems and then come to us as a partnership to apply for funding. And so the funding envelope per project for GeneSolve projects is usually between 50 and 250,000 coming from Genome BC that we ask to be matched 1-to-1. Usually this is coming from the sector partner, and in our new redesigned version, this doesn't have to be cash like it used to be, but it could be in kind. So if, for example, they provide some time of their own staff, or if they provide a platform that you would be using, there could be many different opportunities for what that in kind could look like. And the projects usually could be very short, from 6 months or all the way to about 2 years, depending on the scope of the projects. And so I have put here the timelines for our next intake. As I said, we have 3 intakes per year in the recent, since last year. And for us, we have sort of a 2-step process. First, the expression of interest, and then once that's reviewed and it's ascertain that it fits the GeneSoft application guidelines, then teams are invited to submit a proposal. And then after reviews and all of that, then there is notification of results. As I mentioned to some of you, this is our most, has the highest success rate of all the different programs we're running. We get quite a bit of interest, but it's still, we do have quite a big funding envelope to support researchers who come with projects in this competition. We also have what we call the Genomics Innovation Fund, and this is somewhat of a newer funding opportunity. This is run once a year. So we had one in the summer of this year, and we'll have one coming out next year as well. And so I wanted to sort of bring this up, even though we don't have this open currently for applications, but as you're thinking of the different research projects that you're planning down the line, you could consider this one. So here the applicants could be different from academia, government, not-for-profit, and others. And so the main purpose of this is to support innovation in omics or uses of omics to innovate. So this could be technology development in the omics field, or it could be an innovative way of using omics for any of the sectors that we're supporting. So it doesn't have to be health specifically, but it could be any of the agri-food, natural resources as well. And the requirement is that the projects need to move up 1 level in the technology readiness level scale, and the projects could have up to 250,000 total budget with 1 year to achieve the goals that are set out, and there is no co-funding need for this one. And so we have another project, funding opportunity that we have partnered with Genome Alberta with. This is a somewhat new thing for us. It's called Healthy Outcomes Through Genomic Innovations. It has 2 different streams, but the main thing is that we're trying to support translational research to try to get it closer to implementation if the projects are sufficiently evolved. So again, for this one, the way this would work, because we're partnering with another genome center, that researchers are we ask ideally to have a partner coming from Alberta, and you could apply through them, for example, and then they would put in up to 250,000 of the required amount. And if there is BC researchers like any of you, then Genome BC would put in another up to 250,000. And so there would be still 1 portion of the funds that needs to be co-funded, either cash or in kind from another source. And these would be 6 to 18 months projects, fairly short, and then also need to move up 1 level in the technology readiness scale. Now, this one is a scary timeline. This was fairly recently launched, and the expressions of interest are already due October 21st. So I'm sorry, we're coming to you just a few days before, but I figured I'll mention it just in case you already know of a project that you could potentially collaborate with. So the full proposals are going to be due towards the end of November for those that are invited to submit one. All right, and then there is one last thing that I wanted to mention is there is a Genome Canada funding opportunity that I have mentioned to some of you who I had the pleasure to talking to before this session. And this one, I feel like even if you don't apply for it, you'd want to know about it, and I'll tell you why. So with support from the federal government, Genome Canada has launched this program just last week, I think beginning of last week, called the Canadian Precision Health Initiative. And it has 4 different pillars. I have a couple of slides on the first one, but basically to generate population level genomic data, that's pillar 1, through which they would be generating 100,000 genomes from Canadians across the country. There is the second pillar, mobilizing and advancing the utility of genomics data. So in this case, so pillar 2 and 3 are not going to be launched until early 2026. So if you're interested in any of these, you will have a chance to sort of think about it, start kind of strategizing who to collaborate with on those if you're interested. So the first one is to work with the key users, could be academics and industry who would want to access the 100,000 genomes data to make sure that there is an understanding of what the needs of the end users are going to be, and to make sure that they advance the research. There's new AI-powered methods. And so the fourth pillar is where Genome Canada is going to be assembling an alliance of Canadian partners for genomics in health, so that they can foster collaboration among the various partners across the ecosystem, as well as to align with efforts that are happening across sort of across the country, as well as in regional, provincial initiatives and internationally. And we don't know too much about the pillars 2, 3, 4 at this point because they haven't been launched. But I'll talk about pillar 1 that has. And so as I said, this is going to be focused on generating 100,000 genomes. And so there is applications coming in soon, as you'll see on the next slide. But the idea is that we'll have this national data set that, as you may be aware, that the UK had several years ago, the US is working on, and the countries all around the world are now having, X number of genomes from their own individual countries. And so now the federal government has been putting money into it. So we're excited to have a Canadian national data bank that's going to be generated. And so the purpose is to enable further research and innovation. And so once the data sets are generated within this project, they are going to be going into what they call a pan-Canadian genomics library, a central repository. All 100,000 genomes are going to go in there. They're going to be de-identified, and there is some minimal clinical data that has to go with it. And then there is going to be access to that, or access control, and people can apply to get access to the data and do their own data analytics on the platform. So that's very exciting because I think it's going to generate a lot of cool research findings, and hopefully we'll be able to implement some of those into clinical care as well. So Genome Canada is providing up to $6 million per project. In total, they have a $60 million envelope for this one. They will be asking for the generation of short-read sequencing. They ask for one-to-one co-funding, which could be cash or in-kind. 66.66% of that is going to be provided by Genome BC for BC-based researchers. And the projects could be up to 4 years, some of which is the generation of the short-read sequencing, followed by data analytics. Because each researcher who applies for this, they have a chance to analyze the data, ask their own clinical questions that they're interested in pursuing. And the timeline for this is not a whole lot better than what I showed you for the Alberta and Genome BC collaboration. The letters of intent are due in just about 11 days. with the full applications going in 2 tranches. Those who already can apply in December, and those projects would start in March. And those who need a little bit more time, they have until March to submit the full application. And so hopefully this is of interest to you, even if it's not for the purpose of applying right now, given the very tight timelines. But if you are very keen on being part of one of the teams that are moving forward, please let us know, and we'll try to partner you up with somebody who has sort of been expressing interest to us that they want to be part of this. All right, and with that preamble about Genome BC, now I would like to talk to you about something that I'm truly passionate about, the metadata commons. And for those of you who don't know what metadata means, metadata is data about data. So in the context of omics data, that would be, for example, what was the instrument that was generated on, what was the standard operating procedures that were used, what sample it came from, how that was processed, and so forth. And so when I joined Genome BC, one of the things that I wanted to start up was to know what kind of projects Genome BC has funded, for example, that generated omics data, so that we could let the community know that there is all these wonderful data sets that could lead to additional value. But we wanted to be a little bit more ambitious than just Genome BC funded projects. We wanted this to be expanded to any funding in BC and across Canada and worldwide eventually. And so as we were discussing this, and Annaïck was hired to help with the actual development of the metadata commons, we've been thinking, well, it's all great to have a centralized repository for the omics data sets, or at least the metadata about those projects. But all of these health-related omics data sets rely on samples. And so if we could also include biobanks where samples already exist in various people's freezers, throughout the many different institutions, then that could give a leg up for researchers who are interested in maybe accessing already existing samples to ask different questions. And so we've done just that. And so we developed the metadata commons to keep track of data assets, either omics data sets or the catalog of human biological samples. And one of the things that is part of Genome BC's data science strategy is to support the secondary use of data. And what better way to do it than through a metadata commons, through which researchers could see what data sets are available in the community and be able to use those for purposes beyond what they were developed for. That way we could stretch the research funds that are so limited a little bit further, but most importantly, to derive additional benefits to improve patient care in general. And so, as I said before, we're very big on facilitating new collaborations. So this tool hopefully will lead to additional collaborations between data seekers and those who are custodians of the data, and potentially those freezers full of samples. Maybe the samples have a chance to see the light of day and become data that could then derive additional value. And so we see the metadata commons having many benefits. We listed a few here. Long term, we're hoping that this will lay the foundation for the future of omics research. Most importantly, to remove silos. As I got into the position, and even before having had close to 20 years in translational research, I've been astounded how many silos of data there are, never mind at the clinical level, but even just sort of like knowing whose freezers have what, who has the specific omics data that I would be interested in. So hopefully we could start removing some of those silos to make it more accessible for collaborations to happen. And we really are keen on community building, nurturing a collaborative and knowledge-sharing community. And we're hoping that this will also help change the culture towards being more open to collaborations and sharing. We have, as Annaïck will show you, a streamlined way of access or requesting access to the data through the Metadata Commons. And we really truly hope that this will promote cross-institutional collaborations, across Canada, but even beyond that. And obviously there is an equity component. Not everybody has the funds to develop these very expensive omics data sets. So young investigators, researchers from areas that maybe see less funding, or even just anybody could come and hopefully be able to share in some of the existing data sets. And so you may ask what's in it for you. Maybe some of these are not not going to be of interest to you, but we're hoping that these new collaborations will happen. You'll, each of you will look at it and find some interesting project that they are like, oh, I would love to see if I could collaborate on that, because that's a kind of a cool question that they ask, but maybe I could look at it in a different way. Computational researchers potentially could collaborate with clinician scientists with expertise in the specific disease area, and again, derive further value. Anybody who submits data or metadata rather to the Metadata Commons gets this wonderful data sharing certificate. This is actually our very first contributor, Corbin Bhatti. He contributed the BC Generations Project's metadata as the first project onto our platform. And we also provide a $500 gift card for BC students, staff, or anybody who is submitting a metadata, because we know that it takes time to collect all of that. And so we wanted to have a small token of appreciation for that. And then we'll also have at the end of the year a Genome BC Data Sharing Award for that's going to be awarded to somebody who has provided metadata to the Commons. And as a data seeker, hopefully you can find new discoveries, or if you already have an existing discovery, maybe you'd find a data set through which you can validate those. And we're hoping that it would reduce time to discovery or validation by using existing samples or data. Or you could just go and identify researchers that have expertise in a specific area that you also are interested in. And with that, I'll hand it over to Annaïck, who is going to be showing you the demo of what it looks like. I'll stop sharing that and I'll share the actual site. Let me just okay, just a moment. All right, so you will see. No, it's just because I closed it and it wasn't showing it. Yeah, you can do it here.
So hello, everyone. My name is Annaïck. I'm a Research and Innovation Manager of Data Science. And thank you very much for giving the opportunity to present and to meet with you today. So I'm going to do a quick demo of our Health Metadata Commons platform and a few slides afterwards. So here is the home page designed to welcome the user. The navigation menu is on top. And we have here a dashboard showing an overview of the content of the database at the moment in terms of what sample types, what kind of omics, how many projects per omics, and last but not least, the disease or condition study. And at the bottom of the page, as every page, you can check our terms and conditions listed here. And you can also contact us using this interface. Your feedback is welcome and it helps us to improve the platform. So as mentioned before, the platform is a centralized place where researchers can access or they can contribute. So if you want to access the database, you need to go to navigation menu on top and search. So the projects are listed here and a few information are shown for every project. And the search bar allows to filters down the list of projects. So for example, if you click on the title, you will see the full information about that. So for example, this project, like every project, we organize the information into three main parts: projects, cohorts, and samples and omics. The information we collect about the project is high-level information like the title, the description, the investigators, funders, institution. Is the study complete? Yes or no? Is there any publication and keywords? And also we ask if there is any URL or link to the data access request. The second part is about cohort of patients. So information here we collect is about the size of the cohort, how many patients, or estimated size of the cohort, the study design, enrollment time, and the site, the city or the hospital, and of course the disease, the condition studied. It might be a list like in this case. And we collect also information about biobanking consent, about medical history, ethnicity, and time course. Is it available? Yes or no? If it is available, check the box. That's all we want to gather here. And also some metadata about the phenotypes and the outcomes, if there are any, what kind of clinical data types have been collected, and time course, if it's time course. And maybe this cohort of patients has different groups, could be treatment and control groups. So that's what we mean here. In this case, just one group with inclusion criteria. But if there are any exclusion, that will be listed here as well. And last but not least, samples and omics. So for every sample type, there is a card with information about this sample type. Like for example, here, the serum has some metabolomics. You can click and view more details about it, like experimental design, for example, or what instrument has been used, what the number of samples in this data set. It might be different from the number of samples collected, if not everything went to the sequencing. Going back to navigation menu on top, to another project. And so if you are a data seeker and this project looks interesting, you might want to connect to contact the investigator. So you would click on this button, and this is a template that you can edit, and it will be sent to the investigators without showing the email for privacy reason. So that's the way it has been designed. And last but not least on this page, you can, if you are logged in, you can save your search criteria. So anytime there is a new project that it's coming in the database, you get notified by email. So, you know, you don't need to go and check. It's automated. You can specify the study design you are interested in, and you can select anything in the menu here, or the disease. Let's say you are interested in any studies about asthma, and sample type, I don't know, and omics type. you select whatever you are interested and get notified automatically. That's one feature. So back to the navigation menu on top, metadata submission. So in this case, the researcher has want to contribute to the metadata commons and has some project to submit. So the submission process has different steps. The first step is to create a draft using this button and then submit. Then Zsuzsanna and I will review, and then it's going to be public and available on the search page. So the start new submission opens up a form that the researcher will need to fill in. That's the same information I showed you before, just empty. And there are some drop-down menu to help you fill out the form. Not all fields are mandatory, just the one with an asterisk. So it's about 30-ish in total. So it could take only 15 minutes if you have, and the minimum required metadata is about 10 metadata fields. So on the navigation menu, we have resources page where we list our terms and conditions, user guide, how to submit in details, step by step, and some frequently asked questions. You can find links to our YouTube channel. We have in the data science team at Genome British Columbia data literacy webinars every 2 months. And this is the one we have done so far in the last 2 years. And so you can just go and watch on YouTube channel through this page. We also list our funding opportunities, as well as data sharing principles like the CARE, FAIR, and OCAP. There are direct links here, and the pan-Canadian strategies in terms of genomics, AI, and health data. And this is another page about biodiversity, because we plan to go to other sectors, not only health data. So at the moment, it's only human-related metadata. But in the future, we plan to have different species and more organisms. So at the moment, we just have this page, and in the future, you will have another form for any environment, fisheries, forestry, wildlife project you might have. So we list here a list of biodiversity projects, as well as links to the Earth Biogenome affiliated projects. So we tried comprehensive list here. And last but not least, you can also provide your feedback using this bubble chat button at the top right of the screen. If you prefer, you could also just contact us here at the bottom. So yeah, this is metadatacommons.ca. If you go to .org, it's the same. It goes to .ca, .net, .org, .net, and .com, right, and .ca. So have a look, and your feedback is really appreciated. So I'll go back to the presentation. There we go. So this is a few slides about our education team at Genome British Columbia. So they're working with healthcare professionals to make genetics, genomics aware. So they have two main objectives listed here about leveraging existing opportunities or co-creating initiatives with the clinics. And examples of projects are listed here, like they have a partnership with Pathways BC to manage the referral process. I will show you in the next slide. They have done a mini education module integrated into genetic test report, a pilot program for genomics education for family nurse practitioners, and a project about pharmacogenomics. So this is a Pathway BC website for healthcare professionals. And on this website, there is a lot of documentation about genetics genomics. So at the top here, this is basically the table of content. So it's about either hereditary cancer, fertility, for example, cardiovascular. It's grouped by categories, and it provides one central and unique place where you can find information about what genetic resources you can refer your patient to. Yeah, so it's a comprehensive listing, and there is also wait times, lots of information, and lots of documentation that can be found on Pathway BC. And that's it, I think. We are opening the floor for questions. Thank you.
So thank you guys both for coming. I'll open up to questions here, and then I think we have time afterwards as well if people want to stay and actually check this out and ask questions about navigating the site. We'll be around. So I'll open the floor here if anybody has any questions for the group.
Ever since you raised the issue of secondary use of data, so the data that's in the Meta Commons, when you go through the application, how does that relate to, like, say, ethics applications? Is it that each project has a different requirement for what the investigator would have to do in terms of an ethics approval, or would you want that done prior to reaching out to you?
We don't have the data, but we have, the description of the project. So the connection is going to be made between you and the project, basically the data custodian. It's up to you if you want to have ethics done before. I would say probably you'd want to reach out to make sure that they want to collaborate, depending on what the consent that they have for the specific project, right, from the participants. So you'd want to have a discussion, figure out if there is an interest on both sides for a collaboration, and then you could go through the REB approval to get,
I'm wondering whether you are or will be able to track like collaborations that happens after the sharing of this information, because I think this is quite important for you to also understand the impact of this platform.
Yeah, so for now, the only thing we'll be able to track whether there was an email sent, but we are hoping to do a survey maybe annually to the researchers and say, we saw that our request went out to you. What happened? Did you end up having a collaboration or not? We do see the emails that the requests come to us as well, and we could, follow up and make sure that, if the request came in, we kind of make sure that there at least was a response. But ultimately, it's going to be up to the researcher to see it. And we're hoping that with an annual survey, we can figure out, once we have more data and more requests come in, perhaps we can do that survey more often. But just sort of planning wise, we were thinking one annual survey to see. Yes.
So when you actually accept data, I mean, if somebody's submitting data, what is the process of curation or validation? Is there any specific process to validate the data? Because anyone can actually send data.
Yes, I mean, so We do try to build in some things to make sure that there is an actual person on the other side. It's not an automated, bot who's submitting random stuff to us. But ultimately, it's the two of us now, and we have sort of a team growing in the background who we do validation. Because we both worked in health research, we could at least sort of flag very obvious things. We won't be able to flag, very sort of detailed things. But in our terms and conditions, we're asking researchers who want to participate to try to be good citizens about it. But beyond that, at the moment, we won't be able to do too much.
There are AI generated data, and people might just, bad actors can actually try to contaminate the database or the repository, right? So,
Yeah.
So it's a huge problem for data science, area. That how do you curate data so that data is consistent, and also it is validated.
I know, yeah. And even though this is only metadata, it's the same issues, right? We don't want to be putting up projects that don't exist. So, yes, I think eventually will be a problem. Now we're having sort of one-on-one relationship with most of the individuals submitting. Eventually you'll be sort of, out in the bigger sort of community, and then it's going to be a lot harder for us to make sure, but we'll keep that in mind because we'll have to deal with it. Thank you.
Have we learned anything from the UK where you said they've had this database for quite a while longer?
So that was for the 100,000 genomes. So they actually have the actual data. And so they have their own processes and everything. We've been talking to the Australia Genomics. We've talked to the US group as well as the UK one to try to see how they've done things, so that we can learn for this one, but most importantly for the 100,000 genomes project, where the actual data is going to be there, and it's going to have a lot more sort of validation. At least for that one, for the 100,000 genomes, it's going to be a little bit easier because those are all funded researchers. And when the samples go to these approved sequencing centers, a copy of that data automatically goes into the library. Right. So there is a way through these approved centers that there's not going to be some random stuff going in. But yes, we're trying to learn from those who have done this for a while.
Maybe I'll just go back a little bit then. It might have been on there when you were going through, but is there... The projects don't necessarily have to have published data then to have the metadata uploaded. They do.
Yeah, no, they don't. Sorry,
So,
no.
Is there? Can you see at what stage the project's kind of at then? If it's, I don't know if people submit this in the planning stage, or if they have to have the data, or if it shows that it's actually published data, or.
Yeah, so we take all of those different examples at the different stages. And we do have a way for the researchers to update their records. So for example, if they have, gotten funding and let's say they already enrolled individuals, they can at least include that information we have collected, from this kind of samples. This is the population. And then they can come back in a year or two when they have generated omics data to add that to it. So we like to sort of work at any stage, and then once they have a publication, they can add that to it too. But we'd like to sort of catalog them early on, because then collaborations could start happening the earlier the better.
So. Well, I will leave the formal session at that, but please do stick around and chat. And thank you, Zsuzsanna and Annaïck, for being here. And then also I have to thank Genome BC for sponsoring the lovely meal that you had today. So
thank you so much. Thank you for watching.